Hello everybody, I am Jarrett Ross, the Genie Vlogger, and welcome back to another Professional Genealogist Reacts. On today's video, I will be reacting to How They Caught the Golden State Killer by Veritasium. Now, Veritasium is a channel I'm quite familiar with, been watching Veritasium for a long time, and in fact, when this video first came out, it was a video I thought about possibly reacting to. Part of the reason I didn't react to it at the time was because I was still actively working as an investigative genetic genealogist with the company DNA Labs International. Not too long ago, I had a few people suggesting I do a reaction to this video, and I thought this would be a perfect time. I'm no longer working in the field, but I have a lot of experience. It'll be interesting to see what they talk about in the video. I did see this video when it first came out a few years ago, but I haven't seen it since, so I don't remember everything that is in it. So I imagine it'll bring up some things that'll be interesting to discuss, especially considering that this has been a field that has been growing exponentially every single year. So there's been a lot of changes likely since this video came out, and I don't remember exactly, but I think this came out when there was a lot of stuff kind of going on in the field in terms of the databases setting up the opt-in and opt-out options. I think that's all I'm going to discuss for this intro. This is an almost 30-minute long video, so I imagine my reaction will be long, so let's just jump into it.
This video includes a discussion of serious crimes, which may be disturbing for some viewers, so I wanted to let you know that up front. In the small town of Visalia, California in the mid-1970s, one of the state's most prolific criminals got his start. He repeatedly broke into houses and stole small items.
6 months later, in 1976, he moves up to Sacramento, and over the course of the next 3 years is moving all across Northern California, committing numerous sexual assaults, over 50 sexual assaults. And then in 1979, he moves down to Southern California, starting in Santa Barbara, and starts killing. And he was known as the original Night Stalker in Southern California.
Variously known as the East Area Rapist and the original Night Stalker, these were brutal, meticulously planned crimes that spanned numerous California counties between 1976 and 1986.
Police believed that all of these crimes were committed by the same person, now known as the Golden State Killer. How was he not caught based on fingerprint evidence?
Well, he didn't leave his fingerprints. He always wore a mask. He always had gloves on. This was an offender that did everything to prevent himself from being caught.
The Golden State Killer has been linked to at least 12 murders, 50 rapes, and a string of burglaries throughout California.
So we supply DNA sequencing solutions to the forensic laboratories that process crime scene DNA.
So I worked with Verogen a lot in my time in the field, but one of the things that was especially interesting with Verogen was while I was at DNA Labs International, we were the first lab to get one of the newest technologies in this field created by Verogen up online and accredited. And that technology was Genealogist, which is basically a DNA kit where it takes much less DNA to be able to sequence a profile, a genealogy profile that we can upload to a database and then compare to others to find DNA matches. So with most cases dealing with DNA for crimes, you're often dealing with small amounts of DNA and very highly degraded DNA, which is a major issue. And so having a test that can get a viable profile using much less DNA makes it so that some cases that previously we couldn't even get sequenced, now with Genealogist, we could get sequenced. So when I was at DNA Labs International, we were the first company to get it up online and accredited. I was the first genealogist to provide an investigative lead to solve an unknown human remains case, which ended up being the remains of Kenneth W. Heasley, but a large part of that was through doing the Genealogist kit, which was done by Verogen. Now, after this video was done, Verogen was bought by another company within the forensics industry called QIAGEN. So now I think they call themselves Verogen a QIAGEN company or something like that. And you see Verogen or QIAGEN or GEDmatch. It's actually Verogen's the company that bought GEDmatch. So if you hear any of those three, they're kind of the same company. So GEDmatch is owned by Verogen, which is owned by QIAGEN. I also think the fact that they're interviewing people from Verogen is a good indication that Veritasium did their research and really looked into this stuff, because Verogen, I don't think, is a company that most people necessarily know about with this industry unless you really have looked into things and have learned about more than most people. So if we were looking at like the iceberg of investigative genetic genealogy, Verogen would be maybe 1 or 2 steps below the surface. So not quite what most people know about, but they are well known within the industry. So good sign.
Crime scene, there's typically less than a nanogram of DNA left, and it's usually degraded. Right, so it breaks down.
Why
DNA,
is there so little at
Just
a crime
like I was
scene?
talking
a
about.
nanogram?
Yes.
That's insane.
Yes.
There's no accident that he stops in 1986. 1986 is when the first DNA case of a murder case is actually solved with DNA. And I think he's following that, and he's saying, you know what I'm doing here, I'm leaving my DNA. I can't do this anymore.
Investigator Paul Holes tracked the Golden State Killer for decades.
He couldn't account for DNA technology, and that really was his big mistake. That was the critical mistake. He left his DNA all over California. And it turns out that 3 of the cases, I had 3 sexual assault kits, and that's where I got the Golden State Killer's DNA from Northern California.
So one of the things that is the disappointing part about a lot of older cases is before they knew about DNA, they didn't hold evidence properly so that we would have good quality DNA. So a lot of the cases that you hear that are, especially before the 1980s, especially that 1986 year with the first cases being solved with DNA, well, then you're basically hoping that somebody took the time to store it in a way where the DNA could be held right. Because there's a lot of issues when you're dealing with evidence. A big one that we obviously know about today is contamination. So contamination from other people at the scene, contamination by the people handling the evidence. And this is one of the reasons why when law enforcement is sending their evidence to different labs, these labs have to have proper accreditation in place so that chain of custody stays proper with the evidence. If chain of custody isn't held up with the evidence, then that gives reasonable doubt in the sense that something could have happened where that was changed and now the DNA is incorrect. Whether that be something on accident or whether that be something that maybe someone purposely tried to do something. So whenever you go to any of these labs that are dealing with evidence from an actual case, they have to be signing things in, they have to be signing things out. There are time logs on how long things are kept out. They have to be kept under certain conditions, certain temperatures. When you're going into the labs, there are certain protocols so that you don't contaminate things. It is a very, very serious thing. That evidence has to be handled by labs that have the accreditation that can then do the extraction of the DNA. So the company I worked for, DNA Labs International, they were a company that was accredited. They had everything in place so that they could keep proper chain of custody. So for a lot of cases that you heard about for a lot of these other companies, Parabon was a big one. DNA Labs International was the company that did the DNA extraction. And if you actually Google Parabon and DNA Labs International together, you'll find a lot of cases listed where Parabon did the genealogy, and then DNA Labs International was doing the extraction, especially in the early years. So I joined DNA Labs March 2019 as a contracted genealogist, then I became full-time later in that year. I think it was September, October 2019. But with Parabon, I think they had really gotten started much earlier in 2018, I think, was when CC joined. And by CC, I mean CC Moore. So for those who aren't familiar, CC Moore is probably the most recognizable investigative genetic genealogist, especially because she's been a very recognizable genetic genealogist for decades. All right, enough rambling. Let's keep watching the video. Hopefully I finished whatever thought I started.
But having the unknown killer's DNA wasn't enough. They needed to match it to a known sample. In 1990, the FBI started work on what would become a national genetic database. It mainly stores DNA profiles from convicted criminals and persons of interest. It's called the Combined DNA Index System, or CODIS for short. In each of your cells, there are 23 pairs of chromosomes, one from your mom and one from your dad. And at particular places on some chromosomes, there are short sections of DNA that repeat.
Short tandem
like
repeats.
AATG, 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 and so on. These are called short tandem repeats, or STRs, and different people have different numbers of repeats. Forensic labs produce STR profiles by counting up the number of repeats at each location.
This is doing an amazing job of explaining this, because this is exactly how we'll see it when it comes out. What's the location? How many times is it repeating? And then we can use that to identify very close relations. So only someone up to basically a first degree relation, basically meaning we could identify you to yourself, or we could match you to a parent, match you to a sibling, or match you to a child. There are cases, I think, that have gone beyond a first degree relative, maybe like a second degree relative. I doubt third degree relative, but I know that that is something that a lot of people have been working on in terms of trying to be able to use STRs to be able to get a better idea beyond just a first degree or self-identification. Because obviously, if we can identify more distant matches, which is what makes genealogy so amazing, then we can build much more, which I'm pretty sure they're going to get into. So let's just watch what they say.
Initially, there were only 13 places on the DNA where STRs were counted, but in 2017, that was expanded to 20.
It feels to me like that's not that much information.
It isn't.
But the chances of two people having the exact same number of repeats at all locations is incredibly small.
Now, what's really funny about the probability is when we are dealing with the reports to actually say what is it we're seeing with the DNA, and they're trying to say what's the probability that this DNA is actually from this person, because they're not going to say yes or no, they're going to say a probability. So what they use is what's called probabilistic genotyping. So whenever I was dealing with reports that had the probabilistic genotyping, I always thought it was a little funny, just because these numbers that they would be showing were numbers I'd never heard of. You know, this is a chance of 1 in 6.9 septuplillion sort of thing. So you can see here at the bottom, probability of the STR, 7.62 times 10 to the negative 25. Well, basically that's kind of saying that same thing, except we would have to kind of write it out. And so I would see all of these crazy numbers, because sometimes when we were doing these comparisons at DNA Labs International, they were using what was known as star mix or STR mix. Basically, what this was a machine that allowed you to take a sample that was a mixture of multiple people and then be able to create a probabilistic genotyping result showing what's the likelihood that 1 or 2 or more people were part of this sample of multiple people. And there were limitations on how many people could be within that star mixture, but basically you would get some crazy numbers, especially when maybe it was like a mixture of 4 people and we had 3 different suspects where we had STR standards, meaning that a swab was taken from the suspect, so we can use that as a standard comparison for this suspect. So we would take these STR standards from the suspects and then have it compared to this mixture of possibly 4 people or 5 people. You know, it could vary because sometimes you don't know how many, you can only get an idea. But then using that, you would come up with the probability of how probabilistic is it that it's this first suspect and not the other 2 coming from this? How probabilistic is it that it's 2 of the suspects? How probabilistic it is 3? And how, you know, basically every single combination you can think of. So basically we could figure out who are the suspects who are most likely connected to this crime. And I imagine a lot of people are wondering, you know, why would I as a genealogist know stuff about this kind of stuff? Because this is outside of the genealogy side of the world. And a big reason for that is because during my time at DNA Labs International, I was also working on the administrative team. So I was dealing with a lot of cases outside of just the genealogy world, which gave me a really great in-depth view and understanding of everything going on within that company, as well as the larger picture of what's going on within the forensic world. But all of this stuff that we're discussing right now, this is all stuff that has to be done before they can get to trying genealogy for cases. So they have to get an STR profile from the evidence and they have to upload it to CODIS, and then there has to be no matches against CODIS. So when they see a match in CODIS showing that it's either that person or a first degree relative, then they can take that as an investigative lead and move forward. But if they can't find any matching, then that's one of the things that needs to be done before they can move on to genealogy.
The CODIS database now contains over 18 million STR profiles.
Golden State Killer's DNA profile has been up at the National DNA Database that's run by the FBI called CODIS since 2001 and has been searching ever since with no hits. We did Interpol searching, trying to search other countries' DNA databases, thinking, well, maybe he came from out of the country. That's why, after all this time, we have yet to be able to identify him.
The Human Genome Project was completed. Since 1986, scientists had been mapping and sequencing the 3 billion nucleotides contained in the human genome. It's anticipated that
Soon, private companies began offering genetic tests directly to consumers, and they provide far more genetic data than what's in the CODIS database. I traveled to Houston, Texas to visit Family Tree DNA headquarters.
Now, they call it Family Tree DNA, but Family Tree DNA Labs is actually a company called Gene by Gene. I imagine we may see that at some point because Family Tree DNA is just kind of a part of Gene by Gene. But Gene by Gene is a company that is very big in the investigative genetic genealogy space. For one, the Family Tree DNA database is usable for law enforcement for this investigative work. Not all the consumer databases are, but not only that, but Gene by Gene also does the creation of profiles for genealogy for law enforcement. So law enforcement can send in the extracted DNA from evidence and have that sequenced into a profile for genealogy comparison. So like I mentioned before, that chain of custody, that evidence needs to be sent to a lab that has the proper chain of custody in place, and they extract the DNA. Well, once you've extracted the DNA, now you've just got a thing of DNA, so it needs to be sent off to be sequenced to create these profiles. So at labs like DNA Labs International, they were doing the STR profiles, but they didn't have the technology to do what's known as the SNP profiles, which I think is what we're going to talk about in a minute, single nucleotide polymorphisms. So before they were talking about the whole genome sequence, all these nucleotides, that's the A, C, T, or G in your DNA, every single spot. So that what we say is base pairs. I imagine they're going to get into that, but the main thing here is that Family Tree DNA, owned by Gene, these companies are very big in the investigative space.
There you go.
Thank you. These samples, what are they?
So they're cheek cells. They just scrape the inside of their cheek, they put a cap on, and they send it back to us.
It's like a robot ballet.
And if you've done Ancestry DNA or if you did 23andMe, you did not have the cheek swabs, the buccal swabs, buccal swab. Instead, at Ancestry and 23andMe, you spit into a tube. So that's a big difference. I know some people are always like, wait, you did a swab? Wait, you did the spit? MyHeritage and Family Tree DNA do the swabs. 23andMe and Ancestry do the spit.
Yes. So for DNA extraction, we need to remove all of the protections that are on the DNA and just separate it by itself so we can do testing on that.
Instead of looking at 20 places on your DNA, these tests examine 700 to 800,000.
The Family Finder test is based on what's called a microarray. And this is what a microarray looks like. Each one of those boxes is an individual. We can fit 24 individuals on here. And within that box is about 710,000 positions of your DNA
Those
that we're
SNPs.
measuring. We're getting data for.
710,000.
Yes, and they're called SNPs.
So hang on, that's like 710,000 individual bases, like ATGC,
Yes.
for each per.
Actually, I wouldn't even call it individual bases. I would call it base pairs because each SNP, it's not going to be just one base. It's going to be the base pair. So when you see one spot, one SNP, it'll show you, is it AA, AT, TT, is it C, CG, GG, whatever. That's what it's going to see. So it'll be base pairs. And that's actually one of the difficulties when they're doing the DNA testing is once they've identified all the base pairs, then they have to figure out which each base pair, well, there's two in a pair, right? Well, one came from mom, one came from dad. Well, they have to figure out which SNPs came from mom and which SNPs came from dad to be able to then sequence them together so that they can then compare properly. And sometimes they don't properly sequence them, or what's called phasing. They basically phase it, so you phase the DNA you inherited from your mom to one side, you phase the DNA you inherited from your dad to another side, and then you get an idea of how you match up to people. But sometimes they might take some SNPs from mom, and they might take some SNPs from dad and not realize that these are SNPs from dad and these are SNPs from mom, and instead they think, oh, these are all SNPs from one parent. Well, now all of a sudden we have this sequence of SNPs that is not correct, because it's taking different SNPs from mom and different SNPs from dad. And so when it compares it to someone, it may match up, but it's just an error happening because it's a phasing error, basically. And so this is one of the errors that can happen in the DNA phasing. And not just that, that's after the fact of there's already a 1% margin of error when they're doing that microarray SNP chip. So the chip she was holding, they're testing, she said 710,000 SNPs at every single different consumer company, Family Tree DNA, MyHeritage, Ancestry, 23andMe. They're all largely doing about 700,000 SNPs, but it varies. So they all largely do about the same SNPs, but then each one has SNPs that only that company is testing. There's some SNPs that maybe only a few of the companies are testing, but not all of them. So you get different SNPs tested sometimes with different companies. And on GEDmatch, where you can upload your DNA, there is a tool called the Super Kit, where you can upload all of your different kits from the different sites you've tested at, and then it combines them all together so that all of the unique SNPs that were only tested at 23andMe, or only tested at MyHeritage, or only tested at Family Tree DNA, well, now all of a sudden, instead of having your 700,000 or so SNPs, now you might have over 800,000 SNPs because of all of the unique SNPs that the other companies are testing. Now, I don't know if that's the exact difference of number between the different tests, just kind of generalizing, but that is kind of how it is.
The human genome consists of 3 billion bases. All people share about 99.9% the exact same DNA, which means, on average, only about 3 million bases differ from person to person. These individual letter changes are known as single nucleotide polymorphisms, or SNPs.
So on there, are there a whole bunch of like little pieces of DNA then?
Yes. So what we have is a short piece of DNA that is specific to one SNP. So it's one position in the genome where we know there can be a difference.
Okay.
The results of your 710,000 SNPs can tell you about your geographic ancestry or possible medical conditions, but they can also quantify how related you are to someone else. The more closely you're related to someone, the more DNA you share, and the longer the blocks of identical DNA are. But to compare the DNA of two people, you don't need to read every letter. The SNPs are sprinkled throughout the DNA roughly every 2,000 bases. So if you find long sections where the SNPs match up, you can bet that the DNA in between is identical too. By comparing hundreds of thousands of SNPs sprinkled throughout the DNA, you can see where these identical blocks start and stop, and so how much total DNA is shared.
Everywhere that is highlighted in blue is where I share DNA.
With your father.
With my father, which is exactly
Kind of a weird spot to pause, I guess, but basically I just want to say, as soon as I saw it, that's a parent matching, and it looks a little weird because you can see some spots that are gray and a lot of it that's blue. The gray spots are really the spots that they aren't testing on these DNA tests. And they basically show that, but then they'll show everything else. And so when you're matching against a parent, remember I was talking about base pairs. Well, every spot on your DNA, you're getting base pairs, and one of that pair comes from mom and one of that pair comes from dad. But when they're showing you these results, they're showing you these results not considering whether you have full identical regions or half identical regions. By that I mean, are you matching on one part of the base pair, or are you matching both of the base pairs to somebody? Now, GEDmatch, you can do a one-to-one comparison, and they will show half identical regions, full identical regions, and then the no identical regions. Whereas when you look at a chart like this from Family Tree DNA, this is just showing you half identical region. Well, if you're only going to be looking at, are you matching on half identical, it doesn't matter if it's full identical or half identical, well, when you compare yourself to your parent, Well, you're going to match them on every single spot of your DNA, because every single spot of your DNA, you're getting one of those base pairs, one of that pair from that parent. So it comes up looking like that. So as soon as you get beyond a parent-child relationship, that's when you start to see more segmentation within the DNA. So comparing to a parent with these, this is what's called a chromosome browser, where you can actually see your chromosomes, and then it'll show you where you're matching this person. Well, comparing a parent to a child, it always is going to look like this. The only difference will be at the X at the bottom. And that main difference will be, first of all, if it's a female, they're going to have two X's, one X coming from mom, one X coming from dad, but they're going to show it as a single line, as they do here. So for women, it's going to be a half identical sort of thing. For men, they're only getting an X chromosome from their mothers. And so they have only X chromosome DNA from mom. They get a Y chromosome from dad, no X chromosome DNA. Whatever we're looking at here, we know for sure, because of that X chromosome DNA match, this is not a son being compared to his father. So these are like little things that are very helpful in genealogy, where knowing the facts of inheritance can help us in analyzing the DNA match to understand how are they going to be related to us. And this is the kind of stuff we use for investigative work, and the same exact kind of stuff we do to solve adoptee cases or cases of unknown parentage for whatever reason.
Exactly what you expect, because I should share DNA with him everywhere, because he gave me 50% of my DNA. So this is a second to third cousin. I have no idea who this person is. And you can see here, we are definitely not as closely related as me and my dad, because where the blue is, those are the only regions of the chromosome where we share DNA.
We all know that these genetic tests can help identify our relatives. But the question is, can you do the same thing with law enforcement databases? Can you identify family members using the CODIS database?
You can, and it's only limited to really first order and second order relatives.
That means if I'm looking for the Golden State Killer, I'm looking for either his father, his brother, or his son. We did that over and over and over again with no hits.
Why is it so limited?
Because you've only got 23 loci. If you want to do proper kinship analysis, you need to have more points assessed on that genome to see how related you really are.
Law enforcement is not allowed to search the databases of 23andMe and Ancestry.com, but luckily for investigators, there was another way. An independent website called GEDmatch had been set up to allow people to connect with relatives. You can upload the raw data from any of the big genetic testing companies and search for matches.
So when we initially search the GEDmatch database, our best hits come back at third cousins, which means they shared, our top hit, I think, shared about 1% of the DNA. It's very little DNA that's...
They're definitely cutting out the jargon here, because when it comes to genetic genealogy, saying about a third cousin is not something genealogists really like to say when talking about what a match is. They want to say what's the amount of centimorgans. So that cut right there where it goes, here, I'm going to play it back.
Our top hit, I think, shared about 1% of the...
See, shared about 1%. I bet you anything he says shared about 70-something centimorgans, because that's about 1%. And then they cut that out because centimorgan is very jargony. And one thing I've learned in YouTube, the more jargon you say, especially without explaining it, the more difficult it is for people to follow along, and so people just don't. But with centimorgans, it's just a measurement of DNA. So when you say, I share this many centimorgans of DNA with someone, it's just to give an idea of, okay, well, how close could we be? And using that information along with various different tables and guides that have been created to understand what correlations of relationships at certain centimorgans relate to what relationships, that's what we use. So the main one is the Shared Centimorgan Project, created by a genetic genealogist named Blaine Bettinger. A lot of big genetic genealogists have helped with it in various capacities, whether it be reviewing, submitting DNA, or helping in other ways. This is a massive project which makes it very easy to go in and see if you're sharing 74 centimorgans of DNA with someone, which is about 1%, well, then how much of a relationship distance are we possibly looking at? and that's how we start searching. So a little unfortunate they may have cut that out, but makes sense because there's a lot of things they're talking about, and cutting something out like, what's a centimorgan, it's just something that doesn't really add much to it unless you're trying to learn how to do this kind of stuff. So saying just a percentage of DNA definitely makes more sense, but I guess I just wanted to mention that.
And theoretically, by taking multiple of these individuals that share DNA and building their family trees using traditional genealogy and public source documents like census records and obituaries and newspaper articles and findagrave.com.
And Facebook and Twitter and white pages and Spokio and been verified. And there's all sorts of stuff that we're using. I mean, basically, whatever is available in public, that's what we're going to be using to try to build these trees.
You build this family tree back until you get two of these relatives of the Golden State Killer. You get them to intersect, where now you see, they share great-great-grandparents together. Theoretically, Golden State Killer is a descendant from those same great-great-grandparents. So once I identify that common ancestor, it's now identifying every single descendant from those great-great-grandparents.
So there was a little bit of a simplification here because he kind of skipped a major part of this type of research in that he didn't talk about clustering. So when we first get the DNA matches, one of the first things we usually try to do is we try to cluster them together. So he talked about, when you build these multiple matches and then you find a connection. Well, first we're clustering them together because the clusters we create, those are the people that presumably should have shared ancestors. And once we identify those ancestors, it's not necessarily that the suspect's going to be a descendant of those ancestors. It's just, they're going to be within that family. So once you've identified an ancestor of one cluster, and then you've identified an ancestor of another cluster, that's when you start to do the descendancy research, which I think is what they're going to talk about. So basically, you're building the family trees out and down. So you're finding all of the siblings, all of their children, etc. And so what you're looking for between the two clusters where you have identified ancestors, you're trying to look for a marriage between the two clusters, because this is basically trying to put together your fingerprint of a family tree. Because think about your family tree. Only you and your siblings are the people that have first cousins from your dad's side and the first cousins from your mom's side, and the same second cousins, and so on. Now, there are some special situations like double cousins where sometimes there are more people than just you and your siblings that have some of this unique signature, but largely, most people have a very unique family tree to just them and their siblings. So once you find a marriage between two clusters, that is when you can say, okay, the suspect is very likely going to be a descendant of that couple. So it can be a very long process, sometimes years to figure this out. But once you find one cluster's ancestors, another cluster's ancestors, and then a marriage between those clusters, that's kind of like the golden ticket to this research, where it's 99% likely probably that they're going to be a descendant, that person of interest, from that marriage between those two clusters. So that's kind of the main thing I think they're getting at. But let's continue to see the way they explain it.
And this becomes a huge process. People in the 1840s would typically have 15 kids. You have to identify each of those kids, and then all of their children, and then all of their children. So now we have this exponentially growing family tree.
How wide did that tree get?
I think we had over 1,000 individuals in that family tree, but we knew a lot about our offender. We were confident he was born between 1940 and 1960. We knew he was a white male. We knew he was 5'8" to 5'10". He's in California. He's up in Sacramento in 1976. He's in Southern California in the 80s. We basically narrowed it down to about 5 males, and then it's just investigations.
Yeah. With a suspect at their fingertips, this spring they followed him to a Sacramento area Hobby Lobby store and took a DNA sample from his car door handle. Days later, investigators recovered another DNA sample from one discarded tissue, which registered a match to DNA evidence left at one of the crime scenes.
So in the process of doing a case with genealogy, Genealogy cannot be used to create a warrant, to have an arrest. It can't be used to make any conclusive statements or judgments or anything like that. It can only be used as what's known as an investigative lead. So in this case, they have it narrowed down to 5 individuals. They then give that list of 5 individuals, or at least I assume this is how they did it. This is how we would have done it when I was working in the industry. We would have taken that list. We would have gone to law enforcement, gone over everything with them, explained here are the 5 people that we're looking at, explain any sort of details that we do have about them, and then it's now up to the law enforcement to then and go do their own investigation to create enough evidence to have a proper warrant in place to then get an arrest. So what often needs to be done is a standard comparison. So we've done that SNP testing with the genealogy. Well, the STR comparisons that were already discussed, that's what's accepted by the courts. So for a genealogy case to be solved, there has to be a standard STR comparison between the person of interest and the evidence that then matches. So what they're doing here is they're getting what's known as surreptitious samples. They're going and getting a sample of DNA from this guy that, because of their investigation, they believe that he seems to be one of the likely suspects. They're going to get that standard, and then they're going to do a standard comparison between that DNA and the DNA from the actual evidence. And so that's what they're going to then use to create an arrest warrant if it matches. So that's why they're going and getting these samples first before saying, oh, well, we have the genealogy stuff, let's go and arrest them, because they can't. And that's, a very important thing, because when doing the genealogy research and understanding the nuances and the possible errors and different things that can be going on that sometimes you just don't know. Maybe you're looking into somebody and it turns out that they were adopted. Maybe their parents were adopted. Maybe their grandparent was a foundling. You just never know what sort of paper trail research that you're expanding upon that isn't actually blood connection sort of thing. And that's another major difficulty in this. But that's a big reason why I think, in my mind, I don't have too many issues with this. I know a lot of people aren't the most comfortable with law enforcement using genetic genealogy databases, but especially having done the work in the industry, I feel that there are proper precautions in place and there is a proper flow of the work to get things done correctly. So let's continue.
Countless investigators have worked it and have failed to solve it. Within 4.5 months, a team of 6 were able to figure out who the Golden State Killer was, and that was Joseph DeAngelo.
After the Golden State Killer was identified, the floodgates opened. And we were seeing every week, not only single killers, but multiple killers, serial killers. Every week were being identified from cases in the 70s and 80s.
Because there's been over 70 cases now that they've solved this way.
At this point, it is well into the hundreds, if not thousands of cases solved, especially if you consider DOE cases, which I don't think everyone always does, but basically unidentified human remains as well as violent crimes. It's been mind-blowing how much this is done, and especially with new technologies such as what Verogen put out, that Genealogist, that's adding to the numbers, because there were a lot of cases that didn't have enough DNA to create a viable genealogy profile. But then with new technology that can use far less DNA, so we were talking about 710,000 SNPs for regular microarray SNP testing. For Genealogist, it's 10,230 SNPs, just over 1% of the amount of DNA, yet we're getting viable profiles that we can solve cases with. So a whole new world in terms of adding on to the investigative genetic genealogy.
This seems like a real paradigm shift.
It is. Multiple law enforcement agencies have said that this is the most revolutionary tool they've had since the adoption of the fingerprint.
One of the biggest hurdles for most agencies to be able to start doing the genealogy is funding, which honestly I think is a major issue for a lot of agencies. The backlog of sexual assault kits all across the country at various different agencies and departments has been a huge story, but a big part of that starting to being solved is the fact that there's now funding for it. So SACI grants, sexual assault kit grants, have become a very major thing. Some of these grants have been allowed for genealogy when it's a sexual assault case. But right now, one of the biggest things for sexual assault kits is just the STR profile being created. Because remember, that's the first thing that needs to be done. They need to create an STR profile to upload to CODIS to then do a comparison to see, can they get a hit to the person themselves, or can they get a hit to a first-degree relative? And a big help with this is actually coming from some of the same companies that are working in the investigative genetic genealogy field. So when I was working at DNA Labs International, they were a company that was being hired by a lot of agencies to then take on the task of getting the DNA extracted and sequenced for STR profiles from all of these backlogs from various departments. But like I said, a major issue with most of this stuff when it comes to these law enforcement agencies is just the funding. They need to have the money to be able to have the work done.
Each person who uploads their information to a database illuminates the identities of hundreds of other people around them: parents, siblings, aunts and uncles, and cousins. A study by Ancestry DNA found the average person in the UK has 175 third cousins. And it doesn't stop there. The DNA in one person will be shared by hundreds of people yet to be born. Children, grandchildren, nieces and nephews, and cousins once or twice removed. Realistically, each person shares identifiable DNA with nearly 1,000 people, past and future. So it doesn't take many people uploading their data to reveal the identities of everyone living in a country. Back in 2018, a study found that using an existing database of just 1.3 million people, they could identify 60% of all Americans of European descent. And they estimated with a database containing just 2% of the population, you could find a third cousin or closer match in 99% of cases.
DNA storage is one of the things that we have that is really unlike anything else. If you look inside, this is at minus 20 degrees Celsius.
That keeps the DNA good for a long time?
We have DNA samples in here that are probably about 15 years old.
How many samples are stored in there?
Just over 2 million.
Whoa!
Yeah. So the capacity is just over 2 million.
2 million people's DNA stored in this one small room.
Correct.
So I first did my DNA test in 2013, and I tested through Family Tree DNA. I did 3 tests with them. It was them testing the autosomal DNA, so what we're talking about in this video, the autosomes, the SNPs of, the 710,000 SNPs from the microarray. But then they also are testing the Y chromosome as well as the mitochondrial DNA. So the Y chromosome tells me my father's 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 line all the way back to Y chromosomal Adam. And then mitochondrial DNA is telling me my mother's 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 line all the way back to mitochondrial Eve. So basically it's telling you different things about your ancestry by looking at different types of DNA. But based on that information, I'm guessing that my DNA is somewhere in this lab stored. I'm sure if they said, would you like us to store it? I'm sure I said yes, because that, I'm the type of person that I'm like, yeah, go ahead and store it, because who knows what down the line they may be able to offer that is easier to have if they have my DNA there. But being that was 2013, it's now 2024. My DNA is 11 years old. And so, yeah, mine must have been one of the somewhat older ones when they did this. I guess if this is a 2-year-old video, my DNA was 9 years old, or the DNA that was of mine in this lab was 9 years old.
By 2021, over 30 million people worldwide have taken a direct-to-consumer genetic test, the majority of which are through Ancestry and 23andMe, which do not currently work with law enforcement. But Family Tree DNA does.
If you ask Americans, should we have a national database where everyone's logged in it?
As their DNA.
By their DNA and their name, most Americans will be somewhat uncomfortable with that, or potentially very uncomfortable with that. But we're kind of sleepwalking into that scenario.
There are some people who are concerned with privacy. If anyone chooses or decides that they do not want their results to be used for law enforcement matching. They can remove themselves from that.
I do worry about genetic information being used to discriminate against someone.
My biggest thing is health insurance. I worry about if you have somebody's DNA profile and some law is enacted, gets into the wrong hands, and they say that this person has a proclivity, they're headed towards Parkinson's, they're headed towards this, they're headed towards that, and then rates skyrocket. That's the biggest place that I see as a privacy concern.
I would say that's one of the biggest concerns I hear from people in general as well. At the moment, in the US, there are laws, the GINA law, basically what it says is that you cannot be discriminated against medically and for insurance and a few other purposes using your genetic information. But let's say you take a DNA test and it shows that you have variants that have a high correlation to having a certain type of cancer. Well, the insurance companies can't say, well, because you have this DNA, we're going to bring your rates up, which is basically what he's saying that is a big worry, which I think is a big worry for a lot of people. And, you know, with laws, you just never know how it's going to go, how it may change, whether it be legislators changing it, whether it be the courts changing it or whatever. So who knows in the future, but at the moment, there are laws in place so that that kind of stuff doesn't happen. For me, the biggest thing that I've recognized with DNA is that I don't see any major issues at the moment for people doing nefarious things with your DNA. I, don't know everything going on in the DNA world, so I could possibly be wrong, but I think that goes into the main thing that I always think about is, while I don't see any major issues today of what people could do nefariously with your DNA, that doesn't necessarily mean that something could happen in the future or near future that does all of a sudden make it so that having your DNA maybe is a major issue. We just don't know. I think it is a good thing to still keep in the conversation, because if some of those things may arise, we do want to keep our eyes out for that, so that if something may be happening, we might be able to get in front of it to stop it. I think one of the things I hear all the time from a lot of people beyond the genetic insurance stuff is the idea of, well, are they going to target us with diseases and stuff? Like, a lot of people are really worried about China getting diseases created off of our DNA and then targeting Americans and things like that, which I don't know. I don't think that's something that they can do, but who knows?
Once your genetic information's out there, it's out there. It's not like a credit card where you can get a new one. Yeah, you're giving away something that you don't know what it can do.
Well, yeah, our information isn't ours in the first place.
Right.
Our information is shared.
Yes, with your relatives.
Yes.
And although you may be adverse to this sort of thing, if they've already done it, in a way, you're out there.
The other thing to think about is something we've already discussed, and that if somebody really wanted to get your DNA, it is not that hard. The surreptitious sampling that law enforcement do all the time, waiting for you to throw out a cigarette, waiting for you to finish a meal at a restaurant and then going and grabbing your fork, they can get your DNA. Someone can get your DNA if they wanted to. It'd be very, very difficult for you to make sure every single thing you use that could have your DNA is kept within your person, and that, is just discarded in a way that they could never test your DNA. I think that's almost near impossible. Now, that's kind of a weird thing to mention in this sort of thing, because I think a lot of people are just worried, like, well, you do your DNA, it's super easy to access, and that's the scary thing, is easy access to tons and tons and tons of profiles, which, like I said, if something nefarious may be able to be done with DNA, maybe that is a worrisome thing, but at the time and place, with my understanding of DNA, I don't see anything like that becoming a major issue. But at the same time, I still acknowledge the fact that I could possibly be wrong, because we just never know what the future may hold.
In society today, you very clearly have two camps. There are those that are very comfortable having their genome available to be able to be searched for law enforcement purposes, and there are others that are not comfortable at all. And that's okay.
Right now, Family Tree DNA's policy is we will only accept samples from law enforcement under certain circumstances. It has to be a sexual assault, identification of remains, DNA recovered from the scene of a homicide. And we also do child abductions. So if there's a child that's kidnapped and there's DNA evidence, we will process the DNA so they can try to find out who the abductor is. Those are the only cases that we will accept.
Got it.
Unfortunately, now GEDmatch has flipped off the switch. And GEDmatch has actually said that you're not allowed to have your information searched without opting in.
Right.
Was that decision by GEDmatch a direct result of this Golden State Killer case?
No, it wasn't.
And there was an individual who broke in and assaulted her.
But it wasn't a sexual assault, but it's a serious crime.
I believe tried to strangle her and left her for dead. I believe he thought that, she was obviously dead and left. The police, I believe, approached GEDmatch because that they were concerned that he was going to recommit other crimes.
But GEDmatch's terms of service said they would only help law enforcement in cases of rape or homicide.
Her problem was that she didn't die. If she died, it would have fit the conditions, and they would have uploaded the sample because it met the terms of service and everything.
So what happened? They didn't take that case?
They did.
They subsequently caught the individual from that search that they performed on GEDmatch.
But people within the genealogy world kind of got their passions inflamed, saying that is stepping outside of...
So this blog they're showing, this is the legal genealogist Judy Russell, a very big character within the genealogy world, and she has been a very vocal person when it comes to investigative genetic genealogy. Some things that she's discussed I don't fully agree with, some stuff I do agree with, but she's coming at this from the standpoint of having worked as an attorney, I think it was. I forgot exactly. Very interesting person to look into if you're not familiar with Judy Russell. Her website does go over all sorts of really interesting stuff, but this has been one of the big things that she's discussed. And I want to say that she's been one of the genealogists that has leaned more towards less investigative work with law enforcement and kind of a much more restrained use of investigative genetic genealogy.
And they ended up putting a lot of pressure on GEDmatch.
They flipped the switch because they got freaked out.
But at the end of the day, it was a violation of terms of service. And I think ultimately led to a good thing. Everybody got zeroed out, and then everybody gets to sign back in.
So what they're talking about is the opt-in, opt-out option from GEDmatch, which Family Tree DNA has enacted as well. And basically what it means is that when you put your DNA into GEDmatch's database, you can either opt in for law enforcement to be able to search their cases against your profile, or opt out. So you can still enjoy the access of GEDmatch, but not have to worry about law enforcement accessing your DNA. Now, since then, I don't know when exactly they made it how it is today, but nowadays for GEDmatch, when you're investigating an unknown remains case, so a DOE case, that can be searched against the entire database. It doesn't matter if you're opted in or opted out. You can be compared to an unknown human remains case. For opt-in, opt-out, that is what applies to actual criminal cases. So the violent cases, the homicides, and the sexual assaults, those are the ones that the opt-in, opt-out counts for. But on Family Tree DNA, it's not the same, because Family Tree DNA, if you're opted out, you cannot be compared to any law enforcement cases, whether it's a DOE or unknown humans remain case. If you're opted out, you're just not going to be compared to any. If you're opted in, you're going to be compared to all of the law enforcement cases uploaded to the Family Tree DNA database. But at this time, those are the only autosomal DNA databases available for investigative genetic genealogy, although there are other databases out there that allow mitochondrial and Y-DNA database comparisons. And I guess there are smaller companies that have databases for uploading autosomal profiles as well, but they're much smaller, and I don't know if they're really that helpful or not. Like, I know Othram has their own, which Othram is a big company within the investigative genetic genealogy world.
Subsequently, GEDmatch was sold to Verigen.
I can tell you,
I think that's so funny. I know I mentioned that earlier in the video, but I guess that must have happened right around this time, which I guess, geez, that would have been like, 2021, 2022. And then about a year after that or so, Verigen was bought by QIAGEN.
User today, when they sign up for GEDmatch, about 73% of them are signing up to allow law enforcement to search. We're pretty happy with that.
Yeah.
One of the big downsides with a lot of the issues that happened during the early years of investigative genetic genealogy was that a lot of people left the databases that allowed law enforcement use. So GEDmatch saw a mass exodus of people that were leaving. Family Tree DNA also saw a mass exodus of people leaving their database because they felt well, if they're going to allow these in incursions that go against the terms of service, at this point, what are they going to allow in the future that then I didn't realize that I was agreeing to because it wasn't part of the terms of service? And then another thing with that is that a lot of genealogists were kind of saying, you know, one of the downsides of doing an automatic opt-out for everyone, so they mentioned before, cleaning the slate. Well, there's a ton of people who are in this database that maybe don't log in anymore or have passed away and aren't able to access their DNA because of that. And so nobody can go in to opt them in, even though they may have wanted to opt in. Basically meaning that there are large amounts of these databases that even though law enforcement can access the database, there's just people they're never going to be able to compare to because those profiles are basically just inactive, unopted in profiles.
I mean, that's a high number.
It's a high number.
And it suggests that people want that, in a sense.
How many second cousins or third cousins do you know? You know what I mean? You don't know that many of them.
I know hundreds, if not thousands, but then again, I'm a unique case. So, you know, it's pretty crazy. But yeah, I mean, I know a ton of my second cousins and I know a ton of my third cousins. So, you know, remember with third cousins, that means that you share second great grandparents. But then remember, you have 8 great grandparents. So if you have 8 great grandparents and all of them had 5 or 6 siblings, well, that means that you're looking at like 40, 48 different siblings that then all have their own descendants. And if each of them are having 2 or 3 kids, that's one generation down. And now you're looking at, you know, a couple hundred people. Another generation down, you're looking at a couple hundred people. And then another generation down, which I think is about where you're going to be at. And now you're looking at almost 1,000 people. And that's only if you're looking at 2 or 3 children for each one of those. So you have a lot of those cousins out there. So I know a lot of mine, but I've also been seeking them out.
Without even knowing it, you're helping justice. You're helping a victim's family get a little bit of peace. Not closure, but a little bit of peace.
To get an
Why
answer.
not?
Partly, you could make an argument that if you want to be an activist helping catch criminals, one of the best things you can do is like encourage everyone you know to be tested and put in the database.
Correct. And, you know, there are millions of people who have tested at other companies that are not working with law enforcement. And if they want to help, all they have to do is download their raw data and they can upload it for free into our database.
When I was working at DNA Labs International, this was something that we understood as a company. And so we had a small campaign that we did that was called the DNA Opt-in campaign. Even after I left DNA Labs International, I still have a lot of my banners as the DNA Opt-in banner because I do very much believe in this work. I do very much believe that people can make a huge difference just by uploading their DNA or taking a DNA test and then uploading or whatever, so that they can then help not only find these criminals and identify them, but as well identifying unknown human remains. So for a lot of people, if you know that you have a missing cousin or relative or something, if no one in your family's done a DNA test yet, it's very possible that this cousin of yours is one of the hundreds of Doe cases currently being worked, and it's just a matter of you doing the DNA test to identify them. One case that I worked that's a great example of this is the Beth Doe case. This is a case from the 1970s of a girl who was found mutilated and put into different suitcases that were then thrown off a bridge. And she went unidentified for many, many years until we were able to do the genetic genealogy. And when we first did it, we had her in one database and we had some good matches, but it was a matter of building out these family trees. But she came from a Puerto Rican ancestry, and we could tell very strongly from her matches. But then once we got into a second database, well, in this second database, her nephew was her top match. And so now we didn't really even need to worry about the genealogy as much. We were still doing the work, but we immediately get that information to the agency so they can reach out to this nephew. And once they reached out, they were able to identify her. And had the nephew not taken that DNA test, we probably would have been able to identify her, but it would have taken a lot of work, especially considering that Puerto Ricans come from an endogamous population, which endogamy means that people are marrying within only a specific population group for many, many generations. And that then makes everyone who descends from that population group sharing more DNA on average than a typical person would. So when we talk about those charts of if you share this much DNA, that would indicate maybe you're a second cousin or so. Well, if you come from an endogamous population, those charts become less helpful because you can fall outside of those ranges, because what looks like a third cousin in a non-endogamous family may share the amount of a second cousin in in an endogamous family. Hopefully that made sense. Basically, someone might be your third cousin, but they share the same amount of DNA you'd expect more for a second cousin.
I absolutely believe in a person's right to privacy, but I also absolutely believe we have a right not to get murdered or sexually assaulted or be part, having violent crimes committed against us. There has to be an equal balance.
I think another big thing to consider when thinking of that is people think, well, this third cousin did the DNA, so they can identify me from that third cousin. Well, it's not that they can identify you necessarily from that third cousin as much as it's just if they get your DNA and compare it to a database, they will know that person's your third cousin, and then by building their tree, they can identify you. But the fact that your third cousin is in the database doesn't mean that they're going to be able to go and look at your third cousin and figure out what's the DNA your third cousin shares with you. They still have to test your DNA, which is, I think, something that gets glossed over in these kinds of discussions sometimes, just because a lot of people don't think about that. They're just thinking about, well, my third cousin or my second cousin, or even just say your sibling. Your sibling does a DNA test. Well, even though you're going to share about 50% of your DNA with a sibling, that doesn't mean that they're going to be able to look at your sibling's DNA and figure out, oh, this is what you have for your DNA. They may be able to figure out some parts of it because, you and your siblings will have the same mitochondrial DNA. If you have male siblings, all the male siblings will have the same Y chromosome DNA. But otherwise, everything else is going to be about the same or so, because it's coming from the DNA that recombines.
I truly believe that ultimately this tool and all the concerns with it will probably end up in front of the nation's Supreme Court, and there will be a decision. My bottom line message: if you're going to make a decision, if there's going to be laws restricting law enforcement to use it, make sure that you're making an informed decision and not assuming what it is based on its DNA and genetics and law enforcement, because it really isn't what you think it is.
I think that's a great point, and that was one of the biggest worries for everyone working in the investigative genetic genealogy field is how are legislators and courts and other governmental bodies going to react to this, and what type of laws are going to be put in place restricting this where they may not understand what's going on. If I remember correctly, I think it was Maryland that had passed a law that made it very difficult to do genetic genealogy because some of the stipulations in the law showed a very large lack of understanding of what was going on. But that's the main worry, is that people that don't understand these tests or how this work is done, then creating laws and regulations that make no sense and will actually hinder the ability to do this type of work without making anybody safer. And I want to say probably about 2019, 2020, that was when you started to see a lot of various bodies coming together of both people from the genealogy world as well as people from the forensic world, trying to create guidelines and certifications and different things to create standards for the investigative genetic genealogy before legislators and courts can have their way of then manipulating the law. Because it's much better to have it done by people who are informed and understand the work than to have it being done by politicians who, especially if it's in the legislature, may be swayed by forces that we don't want them being swayed by. But I think that's a big worry, is what industries might be able to step in and turn things in their favor, whereas it's not actually going to be helpful towards the greater public.
These are people who are willing to give up their information
yes.
to find their ancestors.
Yeah.
There may be many others who are militantly private, but they don't get a say in the decision of whether they're going to be searchable through that person or not.
True. That's the choice you make. So that, to me...
That's the choice you don't make.
It's a choice you don't make, but it's the choice we have to make as a society, is what's that balance? There is always going to be winners and losers in my mind, right? And I don't want to use losers in the negative sense there, but in a decision, At the end of the day, you have to balance, in my mind, the public safety with the public privacy.
Okay, that was a pretty good video. It goes in depth into a lot of great information. I was very happy that I talked about Genealogist early on because this wasn't something that was very well known at the time. In fact, when they were probably talking to the CEO from Verogen, they were working on that. And that might have been something in his mind when he was talking about very degraded DNA and stuff like that, because you do a Genealogist kit and you can get a viable profile with 1 nanogram of DNA or less. When you're talking about micro SNP arrays, you need 75 nanograms as the ideal amount, and 25 nanograms is like the minimum amount to have a possible viable profile. But with Genealogist, you only need 1 nanogram. Now with whole genome sequencing, you need less as well. I think with whole genome sequencing, you can do it with as low as 10 nanograms. I might be off on that number, but so Genealogist still is making a massive difference in the field. Otherwise, very great coverage in terms of everything they talked about, in terms of the people that he was interviewing for this. The investigative genetic genealogy field is definitely something that has been evolving a lot. And just within the few years since this video came out from Veritasium, there have been a lot of advancements and changes. And I imagine over the next few years, there will be even more, especially as actual boards and certification bodies start to have things enacted where there become more gateways to get into the field, as opposed to now where it's not quite a Wild West necessarily, but there aren't really any barriers in place to kind of check, do these genealogists know what they're doing? Agencies, are they doing things properly? Do they know everything they need to know if they're doing the genealogy work? Because that's one of the big things a lot of genealogists worry about is law enforcement and non-genealogists learning genealogy in a very basic way and then trying to apply that to a case where when it comes to genealogy, and especially genetic genealogy, there is a lot of nuance that takes years of experience to be able to identify and understand. For a lot of us that have been in the field of IGG, we actually spent years doing similar work for adoptees. So before I was in investigative genetic genealogy, I actually worked as what was known as a search angel, and I helped different people who had come from adoptee situations. Some of them were grandparents who were adopted. Sometimes it was cases where it was the dad they thought was their dad wasn't their dad. Now they wanted to find out who was their dad. And so it's the same exact process for that kind of work that we apply for investigative genetic genealogy. There's just a few differences, most specifically being that the cases for adoptees usually aren't dealing with violent crimes and actual victims and, really terrible things. Obviously, adoptee cases can involve some very serious family traumas and stories, but it's not going to always be the case. Whereas with investigative work, well, that's kind of the whole deal. Hopefully you all enjoyed my reaction and a lot of the added information I was able to include in this. And if you'd like to see more reactions from me, be sure to check out this other one right here. Thank you to my Patreon patrons and YouTube members.